DNA is an amazing tool for forensic scientists. It is instrumental in being able to identify biological material left behind by an individual at a crime scene and identify who that material came from. Gel electrophoresis is the final step used in analyzing DNA, and this is the step that enables forensic scientists to visualize the DNA. This process separates DNA based on the size of the DNA fragment. The process is fairly simple and can be done in a high school lab with the right equipment and materials. Prior to beginning, the DNA was extracted from the source. PCR was also performed, which means that the DNA was amplified or copied so that there's now enough DNA that we're able to see it on the gel. During gel electrophoresis, the DNA is injected into a well in a small agarose gel. This gel has a similar consistency and feel as Jell-O. Our first step is to make that gel. This is done by mixing a small amount of agarose with something called TAE buffer. The TAE buffer is used instead of water because it allows us to better control how conductive the gel is, which is important later for when we're running our gel. After mixing the agarose and the TAE buffer in a flask, this mixture is microwaved until it reaches boiling. After the mixture cools, it will solidify into the gel we will use to run our DNA. We also need to add something to the gel that will help us be able to see the DNA. DNA by itself is colorless, so we can't see it if we put it into the gel. There are many ways to visualize DNA, but the safest way to do it is by using something called gel green. A very small amount of gel green is added to the gel after we microwaved it once it's cool enough to handle. Then, the gel is poured into the gel casting system. The gel casting system consists of the casting trays and the comb. The comb is used to create the wells, or small holes, in the gel in which the DNA will be injected. After microwaving our agarose TAE mixture and then adding a small amount of gel green to that mixture, we are ready to pour the liquid mixture into the gel casting system. As it cools, it will solidify. Once it becomes solid, you'll remove the combs from the gel and the trays out of the casting system. The next step is to assemble the gel electrophoresis system. This system consists of a carriage, a buffer chamber, a hood, and a power cord. The gel in the plastic tray that we just made gets inserted into the buffer chamber. The buffer chamber gets put into the carriage, and the carriage is plugged into an electrical outlet. TAE buffer is poured into the buffer chamber until it just covers the gel. This buffer enables us to make sure that there's an even charge throughout the chamber and also better controls the pH than water would. Our next step is to add our DNA samples into the wells. The amount of DNA needed is very small, so we use a piece of equipment called a micropipette, which allows us to measure and move precise amounts of liquids. Our first well is always the ladder, also called the standard, which is a known sample of DNA for which we know exactly how many pieces of DNA there are and exactly how long those pieces of DNA are in base pairs. The ladder or standard is used much like we might use a ruler to help us judge the lengths of our DNA bands in our unknown samples. Samples are micropipetted into the well, with each well containing a different DNA sample. The hood is then placed over the carriage and the gel electrophoresis chamber is turned on. Once it is turned on, there's an electrical charge difference from one end of the gel to the other. Near the wells where we put our DNA samples, there's a slightly negative charge. On the other end of the gel, there's a slightly positive charge. Since DNA has a negative charge, it wants to move to the opposite side of the gel. Smaller pieces of DNA will be able to move faster than larger pieces, and thus we'll be able to separate our DNA samples based on the size of DNA fragments in those samples. This shows a time-lapse video of that process at work. If the gel was allowed to continue to run, eventually all of the DNA would end up at the bottom of the gel, and could even run off the gel if allowed to run for too long. The final gel would look something like this, and enables us to visually be able to see how many pieces of DNA are in each sample by seeing how many bands there are in each sample. We can also use the latter to help us estimate how many base pairs long each sample is. Now, the next time you hear about DNA being used in a forensics case in the news, you'll have a better idea of the process at work behind the scenes in the lab in order to make that happen.